why do you think that it's important to have so much, well, annotation or so many programs for annotation? Why? Sorry?
Programs are complete to annotate correctly.
Yeah. So that's a very good point, but imagine that you get a genome, yeah? And well, so you see that most of the proteins that are in that genome are encoding for hypothetical proteins. We don't know the function, but we don't know the function because there are many databases that are specialized, okay? So we need to combine many of them to get as much information as possible. So for example, here in this plot, you see the annotations of PROCA, yeah? In the y-axis, I don't know if you can see in the x-axis, different tools of annotation. So here is the ratio of hypothetical proteins in the y-axis, and then in the x-axis, you have the different tools of annotation. And then you can see that PROCA has a higher amount of hypothetical proteins annotated when you compare, for example, with BACTA. Okay, so you are getting more information from your genomes. Let's move on a bit into even more specialized databases. I think this is quite important for many of you that are working in so many different topics, but imagine that you have healthy plant and diseased plant, and yeah, so you look at the microbiome composition, and then you find that there are some bacteria that are more abundant in the disease, and these bacteria could be doing any kind of function, right? So how to determine which functions? Well, you go to these specialized tools, and then you see, for example, if they have more chitinolytic enzymes or cell wall degrading enzymes, and so on. So for example, here, I'm showing you the Casey database, that it's basically a database that is specialized in carbohydrate degrading enzymes. At the end of the day, if you are working with, for example, a fungus, it's made of chitin. Chitin is a sugar, and it needs to be degraded. Yeah, so if the bacteria that you are working with that has antagonistic activity or any activity against the fungus, so you could be able to find those genes using this kind of tools. You can build this kind of networks using these tools. So for example, here, what I'm showing you are, this is an annotation based on the Casey database. And the Casey database use HMM domains, just to give you a bit more detail. And well, here we have the healthy and the diseased plant. And what you can see is that, well, when we annotate the metagenome in this case, we have many different types of carbohydrate degrading enzymes. And which ones are popping up? Well, for example, the chitinase, yeah? Or the beta-glucanase or the glycosyltransferase, all related with chitin degradation, okay? So you can see that, well, this is a network. I don't know if you can see the edges, but every node that you see here, every circle or a square, it's a node. It's one gene. And then the connection between them is the phylogenetic distance between them. So we see different groups. The blue ones are the glycosyl hydrolases. The red ones are the non-catalytic carbohydrate binding modules, and so on, okay? So you really can make this kind of graphs and make sense of your data. How many of you are familiar with the antibiotic resistance? Are you working with antibiotic resistance? Yeah, I think you know. So, well, there are, in the last few years, there have been quite a lot of tools for the detection of antibiotic resistance, okay? And you can run your genomes in these tools, but well, not so long ago, there was a publication of this tool harmonization that combines all the tools. So if you are working with antibiotic resistance, you could use these tools to predict your potential genes for antibiotic resistance. Okay, all with me? Good. Do you see this? No. Well, the question is, indeed, why so many annotations? Why do we use so many? Well, this is a table, yeah, of our genome, the genome annotation, the genes, yeah? So in here, in this column, you have all the genes. And then now we have the COG annotation, but also we have the PFAM annotation. And then we get also the KEG annotation. And we can add more and more and more. So you can add all your annotations for every gene of your genome. And then you can extract as much as you can squeeze your genomes in that way. What happens if we have a metagenome? We get pretty much the same. But we get also the taxonomy, because you have sequenced everything. You have sequenced all the DNA from your sample. Okay? What is missing here? Well, if you have a metagenome, you will have an abundance table, right? And a metadata. So basically, you can compare your treatments. You can compare your treatments. If you have healthy and disease, you can compare them and try to find specific functions and taxa that are correlated with your treatment of interest. Questions? Are you all with me here or not? Yes? Excellent question. So the question was if for the same gene, using different databases, we can get different annotations? That was the question. I never had that. That's quite specific, but I am sure that there will be some cases where you get different annotations. You have to think that many of these annotation tools, one works using HMM domains, and some others use clusters of orthologous groups. So basically, you do clustering, like we do with the 16S amplicon to generate OTUs. So it could be that you get different annotations. Maybe it's rare, but it could be. I don't know, I'm looking at Guillermo now. Yeah. Get the microphone. Yeah.
It's not super common, but it happens, let's say, quite a lot. So that's also one of the reasons that it's so important to have multiple annotations from different databases. So then you have the same gene, you annotate it with different databases. So if you see that each database is saying the same, you can trust that. So if you see that each database is saying different things, then you are going to be, I don't know if I can trust that the function of this gene is this.
Yeah. Then if you are interested in that specific gene for any reason, then what you should do is to maybe use a specialized database. I have shown you just a few specialized databases. There are plenty. If you are working with type 3 secretion system, for example, there are a specialized database for type 3 secretion system. So then you should go a bit more in detail in there. Okay, so is it clear? Yeah. So then let's continue. Yeah. So remember this, because on Monday we will come back to this, because, well, imagine the complexity of a metagenome when you get all of this information, KEGG, COG, PFAM, the taxonomy for every gene, the abundance of the genes, then the metadata, and how long can be these tables? Well, millions and millions of rows. Actually, I made a subset to open this in Excel to show it to you, because you need to have Linux servers to open these tables, okay? It can be gigas. Okay, so let's go back to our practical. So, because we're gonna dig a bit more into the specialized database, okay? So we isolated the microbes. We are doing antagonistic assays, and we expect to have something like this. Yeah, so we expect to be able to kill our pathogen and then solve a problem in agriculture, right? That's what we want. Then we have the genome information. So how can we try to predict, yeah, the genes that could be involved in that phenotype? There are many tools. I'm going to show you one of them that we use in our group quite often, okay? And the tool is Antismash. How many of you are familiar with this? No? Never heard about it? No. Nobody? Kumar, yes. Okay. Good. Well, Antismash is a web server and a standalone software to identify, annotate, and compare gene clusters that encode biosynthesis of secondary metabolites in bacterial and fungal genomes, okay? So most likely, the activity that you see here, it's caused by a secondary metabolite. A secondary metabolite, it's, for example, an antibiotic or any other compound that can kill other bacteria, fungus, etc. Okay? So here we have a list of types of metabolites. Okay? So we have, for example, non-ribosomal peptides, or we have ribosome synthesized peptides, and then we have, for example, here the compound that is produced, and well, also the reference. Okay, so this is, so you all know what are secondary metabolites now. Yeah, so basically, thinking this way, it's a compound that can kill the fungus that we want to kill. Yeah, let's leave it there. So these secondary metabolites are organized in what we call biosynthetic gene cluster. So this is the definition of biosynthetic gene clusters. A BGC can be defined as a cluster group of 2 or more genes in a particular genome that together encode, yeah, a biosynthetic pathway for the production of these specialized metabolites. And then here you have a biosynthetic gene cluster. So they can be quite big, yeah, 40, 50, 100 kb. So, and here you have more or less all the components that will be included in these biosynthetic gene clusters. So normally you have the core or scaffold biosynthesis that are these green arrows that I show you. Then you have the tailoring enzymes. When I say tailoring, I mean like enzymes that will take the peptide produced by these 3 genes and will make a decoration. Decoration could be change of a carbon into a COH2 that could end up in being antagonistic against a fungus, for example, okay? So there are also genes involved in the transport because, yeah, it needs to go out of the cell, the regulation for the production, even precursors for the biosynthesis, and something quite important is that it should have also the gene involved in the resistance to this secondary metabolite, Okay? Thumbs up? Excellent. So here I put you again the central dogma for the specialized metabolism. So we have the biosynthetic gene cluster, and then if we have this cluster, we are able, yeah, to synthesize this natural product or this secondary metabolite or antibiotic or antifungal that then we can use it to kill other targeted bacteria or fungus, etc. Okay? This is clear. So this is how we normally think that this is happening, right? You are all familiar with this. So we have the ribosome. The ribosome will read the RNA, and then it will produce the peptide. And then the peptide, yeah, will depend, of course, on the template, on the RNA template, and this is what will be our product, our secondary metabolite to kill the pathogen. Yes? Okay, so what if I tell you that there is another way to produce these secondary metabolites? So there is another way. Are you familiar with non-ribosomal peptide synthesis? Yeah. No? Okay, great. So these are, Josh will expand on this, I think, on Monday. So these are non-ribosomal peptidosynthetases. Of course, they are encoded in the genome, yeah, and you get these small, well, not big proteins. They are kind of a factory because they have different modules. And every module, yeah, this is totally independent of the ribosome. Each module of the protein will add a new amino acid to the chain to produce a secondary metabolite. So basically, it's what you see here. The purple module is adding one amino acid, then the red takes over the purple, and then add another one, and another one, and another one, and another one, until we have a peptide. Okay? So this is something completely new for you, I guess, for many of you. But this is also happening in bacteria and fungus. Okay, so this is the Antismash database. So we can detect all of these secondary metabolites in our genomes using this website. So you can type in Google Antismash, and then you will see this kind of interface. We are now in the bacterial version, but Marnix Medema from the Netherlands, he already created several other Antismash for fungal sequences or even for plants. Okay, so it's quite easy. So you go here, and then there should be upload my genome, and then the genome will go into the server, and you click submit. There are some options that I am not going to go in detail, but there are some options that you can click. You can read about it, and then you will get your output, the result of the Antismash, that it's the prediction of the secondary metabolites. How it looks like? Well, looks like this, more or less. Okay, so you have, this is in a genome, so one single genome, yeah. So, and then it predicts the different secondary metabolites that are produced by your bacteria. So each of these numbers or different colors that you see here are different secondary metabolites or biosynthetic gene clusters. So, and they have different colors because they are from different categories. It can be non-ribosomal peptides or terpenes or ribs or etc. So there are so many. You can look a bit more in detail. Do you see this? Yes. Yeah. So, and well, as I said, you get quite a lot of information with this tool, and it's very interesting for us that we are working with antagonistic bacteria or so on. So, and well, you can dig a bit more, you can click in the gene, and then You can zoom in the region. You can see also the annotation because it's connected to other databases, because this tool contains many annotation tools and many different databases. You can also go a bit more in detail. This is the gene cluster that we saw before, the biosynthetic gene cluster, and then you can see for every domain which kind of amino acid it's encoding. Yeah, remember I told you. So the different parts of the gene, of the factory, this small factory that is an NRPS, will produce different amino acids that will end up being a secondary metabolite. Clear? Yes? Okay. You can also see if there are other homologs for these gene clusters in other reference bacteria. So you run your genome, you detect 15 NRPS, and then you say, okay, well, then, yeah, this biosynthetic gene cluster is already known. Why I'm going to focus in that one? So I should focus in one that it's unknown. Yeah? And the good thing also of this tool is that you can download the output. So you can download this, and then it will open, yeah, all the information, even if you don't have internet connection or whatever. So you can download, and it's quite easy. Okay, do you think that we can do Antismash in metagenomes? I didn't say here, but normally when you do Antismash in a genome, you get 15, 20 different biosynthetic gene clusters. So imagine that you run this in a metagenome. How many clusters will you get? Huh? Too many. Yeah, quite a lot. So if you run Antismash in a metagenome, you will get something like this. Yeah, so this is an example from one of our research, and we got, this was from an endophytic microbiome. So, and we got around 730 biosynthetic gene clusters. So imagine if you do for a rhizosphere, that it's more diverse, you will get even more. Okay? Well, but then, of course, we cannot look one by one. It will take the whole PhD or the whole postdoc looking at one by one. What you do is, again, network analysis. Okay? And if there is time this afternoon, we will build this network together, if there is time. Will you be interested in doing that? Yeah, we are going to try. Let's see. So here, again, a network. It's composed by nodes and edges. So each node is representing a biosynthetic gene cluster. Clear? So, and the connections between them is representing the phylogenetic distance, the distance between them. I'm not going to go in detail in the distance because there are many ways to measure, okay? But just for you to know, this is basically what is happening. So then if they are closer, if they are more similar, then they will be closer. If they are far away, yeah, they will be a bit more separated. If they are totally different types of BGCs, then they will be in different clusters. Is that clear? Good. So then that's what I told you right now. So for example, if we annotate them based on the type of metabolite that they produce, they will be clustering in different groups. So for example, here we have terpenes. Yes? Then here we have NRPSs and PKSs and resorcinol, lantipeptides, lasopeptides. You see the use of this tool? Is interesting for any of you for your research? Okay, I'm happy to hear. Yeah, then even you can go even more in detail. Yeah, I'm not going to go much into this, but you can annotate each of the clusters based on the taxonomy, because as I said, this is a metagenome. Yes? Yes, okay. Okay, so, well, there are so many tools about secondary metabolites. I really encourage you to do a bit of exploration, and if you are interested in this topic, yeah, go a bit more in detail when we give you the slides. So this is the MIB database. So it's an online repository where you have all the known and unknown BGCs. Well, not the unknown, the known BGCs, sorry. So, and the ones that are connected already to a molecule. That's giving a lot of information already. I mean, that's really cool, actually. So, and it has the same shape and the same interface than Antismash. Actually, it's created from the same developers. And, well, you can see the genes and the metabolites, and if there is any compound described, if the genes are known, everything you can see in this one, okay? It has an interface. So this is the interface. Well, this is another tool. It's called Bigscape, that it takes the outputs from MIB and Antismash. We are not going to go in detail, so I will show you this afternoon. So in this interface, you can see the general overview. You can see also phylogenetic heatmaps that this afternoon Guillermo will go more in detail on this. You can see the types of terpenes that were in your sample, or NRPSs, the types of metabolites that are in your sample, okay? And then I want to go a bit more in detail just for you to see what could be the use, yeah, and the advantage of using this tool that combines data from Antismash and from the online repositories. So here you have your BGC of interest, okay, in yellow, for example. These 4 BGCs are in your bacteria, and then you found that are highly expressed in your genome, for example. Let's put that as an example, a genome. So here in purple, we have the ones that are already known. Yes? So what we see is that most likely the BGCs that are highly expressed in your genome most likely will be similar to these ones. So if they are similar, because these ones are already described in the database, in the MIB database, so they are already known. So these ones, for example, yeah, are already out there, and yours are quite similar to this. I would say I don't want to focus on that one unless there is a tailoring enzyme that could give another structure, okay? But then, for example, here in my genome, there is a cluster of other genes, yeah? And these ones are completely unknown. There is no MIB reference, biosynthetic gene cluster. So this one, if I will be doing the PhD, I would say, okay, this one is completely unknown. I would prefer to focus in that one. Are you with me? Yes? Good. This is just to give you an idea of how complex it can become, but yeah, there are so many tools, and yeah, they are all listed here for the secondary metabolism. I didn't explain all of them, but yeah, you can even do phylogenetic inference using Corazon or many other tools and yeah, and possibilities that we have using these methods. Okay? Questions? No questions? Excuse me? That's a next, sorry. That's a very good question. Depends. Oh yeah, no. If there are any tools to detect toxins. During my PhD, I was working with mango toxin. Yeah, it's a toxin produced by Pseudomonas syringae and affects mango trees. That toxin is not detected by Antismash. I don't know if now it's detected, because at the end of the day, you have to think that Antismash it's working with HMM domains that are already known. Okay, so it can predict to sort of level, but if yeah, I don't know if you get what you mean. So if basically if your toxin is not in the database, it's not gonna be detected. Yeah, I don't know if there is any specific tool for toxins. I look Guillermo and Luisa. Yeah, Guillermo wants to say something. Can someone give the yeah?
Yes, that's one of the things of Antismash that is trained with things that are already known. But for example, your toxin, most of the times it's going to be surrounded by genes that are involved in the, for example, the secretion of that toxin. So maybe Antismash will detect it because it will detect that there are secretion proteins around. So maybe it doesn't detect the toxin itself, but it will detect different mechanisms that are associated to the toxin.
Yeah, so then if you are interested in toxins, you could try to find any, because maybe there are tools out there for toxins, I don't know, could be. But yeah, Luisa wants to say also something.
So the only one I know, because I don't work with toxins, I know there's a project going right now, they started this year, on animal toxins. So it focuses on venoms from snakes and things like this, but they're barely starting, and that's what they're trying to do, build a database with toxins so that other tools can use it.
Yeah.
That's all I've seen.
Yeah. Also, you should look at the type of toxin that you have, because there are different types of toxins. Yeah? Any more questions?
So in case of metagenome, is there any tool to filter out only the metabolites from a specific group of microorganisms? Like, to filter out only the functions of bacterial genes? Like that.
You mean to match the metabolites with the genes? I think there are some tools for that. I don't know if they are already out there, but I know that they are under development. I think that's also Marnix Medema, together with Justin, Jos, what's the surname of Justin? I don't remember. Van der Host, yeah. Yeah, they have quite a lot of tools related to that you are asking, so to connect metabolites with genomes. And there is even a repository that we participated in that project where you can already access, but I'm not sure, yeah, how well established is for now, yeah, at this moment, yeah.
Please, can you explain again the non-ribosomal peptide synthesis?
Sure.
And when does it occur in the cell?
Sorry, when?
And when does it occur in the cell compared to the ribosomal synthesis?
Yeah. So Here you have the normal way, yeah? So this is the ribosome, then you have the RNA. It will go through the ribosome, and then you get your peptide, yeah? That's the normal synthesis. So here we are talking about a biogenetic gene cluster, yeah, that it's encoded in the genome. Okay, so then when you get that protein from the genome, it's basically this long protein. It's a huge protein. It's a huge factory. Okay, and then this protein has different domains. That these domains, what basically they do is they produce or they take an amino acid, yeah, to generate a peptide. So each of the domains is specific for each of the amino acids. And at the end, you will create a specific peptide for each non-ribosomal peptidosynthetase. If you see here, we didn't involve mRNA. So we are directly, the protein, it's directly synthesizing the peptide. Yeah? It's not the ribosome synthesizing the peptide. It's the protein synthesizing the peptide. Yeah? Josh will go again on it. Maybe Josh wants to explain in other words.
Next week, Monday, I will also talk about non-ribosomal peptide synthesis because there's a lot of interesting metabolites that are produced through these pathways. The most simple, if you want to have a picture of how it works, the assembly lines in a factory to make a product, that's what non-ribosomal peptide synthesis is. So something, a backbone arrives, at the start of the assembly line, and then first person adds, the seats. The other one puts the engine in, etc., etc. And then all the way at the end, there's always somebody who put the wheels on to make the peptide signaling, right? And then there is also always a person that releases the product from the assembly line. So all these elements, like Vic said, they are contained within this protein with all different domains, and every domain has its function in this assembly line. Maybe also good to mention, Vic, because we talked yesterday about repetitive sequences, right?
Very good point.
These non-ribosomal peptide synthetases, the gene clusters that encode for these proteins that basically constitute the assembly line, there's a lot of repetitive sequences in there because these domains, they are slightly different when you go through the assembly line, but they are much lookalikes, and sometimes they are even the same. Yeah? So one puts in the front seat on the left, and the other one puts in the front seat on the right. I'm just trying to make a picture of this. So, yeah. I also have, in my slides, I have a little YouTube link where you can, there's even a visualization. I always put the assembly line of a car there. I don't know why, but I think it's at the wheels at the end. Yeah. Okay, good. There's more to that, but super important and very difficult to work with because they are super large gene clusters. Those are gene clusters up to 40, 50, 60 kilobase big. So to work with those from a molecular microbiology perspective, super difficult.
Yeah, and think in this way. So this is a bit more in detail what it's inside an NRPS. Okay, so you have different domains, and every domain it's gonna be adding a different amino acid. So that's why the repetitive sequence. So imagine you have here condensation, adenylation, condensation, adenylation, condensation. So they are all repeated after each other. So that's gonna cause a lot of problems and troubles when you are aligning and, well, assembling your genome. Yeah, so normally these are the break points for the genome assembly. Good. We are good? No more questions? Do we want to have a small break or we want to continue? Continue? Yes. Okay, then yes. So I think I have told you already quite a lot of tools. I don't want to overwhelm you with more. But yeah, if you have any specific tool that you would be interested in, like for example, toxins, yeah, well, you can ask me or Guillermo or Luisa just to see if we know if there are out there any tool doing what you need. Yeah? Let's move on a bit to the comparative genomics, okay? And what I'm going to do now to explain you the comparative genomics, because this afternoon we will have more on comparative genomics, what I will do, I will give you examples of comparative genomics. Yes, and then we will go through them. Okay, so we are again in this part in all our flow chart of the annotation of genomes and metagenomes. So now we start with the examples of comparative genomics, okay? But first, I want to start with some definitions that you already most likely know. So you all know what is a gene. You all know what is a genome, I think. Yes. Then you all know what is genomics. Comparative biology, I think you know. And then comparative genomics. So the comparative genomics is basically the comparison between genomes, metagenomes, or even fragments of DNA. So if you have context, you can also compare them, okay? So that's what we are going to do in the next examples. Okay, so I always like to use this slide because here you see how far you can go with the comparative genomics, and yeah, so you can compare so many different taxa, right? So you can go from humans to, well, whales or whatever it is, a fox or wolf, to rats. You can compare all of those genomes and then see what is the difference between them. You can be also more specific, like comparing mammals, flies, fungi, whatever. But at the end of the day, this is what we want to get. We want to know what is lost in some of them or what is gained in some of them to make them different, yeah, for the phenotype that we want to know what is which gene or which group of genes are making the phenotype that we are interested in. Okay, so this is basically what we are going to do. Okay? You cannot read this. No. You can? This is a genome. Yeah, you know which genome it is? We know this guy quite well. Huh? No. Why yes? No. Huh? No. We have been suffering the last 2 years. COVID. So this is the COVID genome. Okay? And these are the open reading frames of that genome. Okay? So just when COVID hit, there was this paper published in Cell, and they were using the different variants of COVID, and they were able to identify several new proteins using comparative genomics. You see how important it is? Because, well, I'm not 100% sure, but maybe doing comparative genomics, you can detect new proteins that could be targeted for the vaccines. So there you go. I think this is one of the most important examples that I could give you. Okay, so you are going to hear more about this. I'm going to give you another example about comparative genomics of something that we have done, well, I did during my postdoc in the group of Joss. So working with disease suppressive soils, you have seen already this picture. Joss will expand a bit more on Monday. Yeah. But here, basically, it's what you have, right? You have the plants growing in suppressive soils and plants in conducive soils. So these suppressive soils are soils where the plants don't get sick or get less sick. Yeah? And then we have the opposite, that is the conducive soil, where the plants get sick. Okay? So then you see that the plants are growing healthy and happy in this condition, and then here they are dying because there is a fungus that is killing them. So in the group of Joss, there have been so many people working in this topic, and I was one of them. And I decided to focus on Betaproteobacteria because it was one of the groups that pops up in the metagenomics analysis. Okay? So when I came to Joss' group, the first thing was to try to isolate Burkholderia, yeah, that belongs to the Betaproteobacteria group. Anyone familiar with Burkholderia? Cool, nice. Is it difficult to isolate? Yes? Yeah, it's difficult. I almost got fired because that, yeah. No, but then the first thing that you try to do, you have a soil, and then yeah, you need to find a way to isolate the bug that you want to target, right? So then I did a literature research, and then I found several media that could help me to isolate this bacteria. I'm not going to go in detail. The efficiency of isolation that target microbe was so-so. Let's leave it there. But at the end, I managed to find several bacteria, several Burkholderia, and they are here in this phylogenetic tree. I think this tree was made with Mega. Long time ago, Mega 5 maybe. So then, well, I could find several Burkholderia, so the ones that are colored were the ones that I found. I found Burkholderia graminis, Burkholderia caledonica, Terricola, Hospita, and some members of Cepacia. Okay, so I could isolate them, and then I did activity profiling, soil bioassays, diffusible compounds, volatile, and so on, so on. So it was quite a stressful time for me because normally we will focus on diffusible compounds, and then you do these antagonistic assays on plate, what you are doing in the practical. And then when I was using all my bacteria, via diffusible compounds, they didn't have any activity. I think you wanted to fire me at that time. Yes, you see? So I spent a lot of time isolating them, and they didn't have activity at all. Then I decided to move on with those bacteria to soil bioassays. Yeah, and this is how we do the soil bioassays. So, well, we have these trays. Do you recognize these trays? No? I think Kumar does. So we call this assay fricandel bioassay. What is a fricandel? You know? It's so nice. You're going to love it. We are close to lunch, so I wanted to show you. So these are the fricandels. It's like a sausage. This actually is the fricandel especial because it has onion. And then we use these trays to perform these experiments. This is very typical from the Netherlands. Yeah? Just hungry? Kumar, are you missing the fricandel? A lot, you see. It's nice. Okay, well, how do we do these experiments? Yeah, well, so we have the control. We have the one that is inoculated with the pathogen, and then we have the one with our bacteria of interest, okay? The one that we think that could potentially protect the plant, right? So, and then where do we inoculate the pathogen? We inoculate here in this spot, here, you see, and here. And then we have 16 seedlings. I didn't say, but we work with sugar beet. So you grow your sugar beet seedlings, yeah, and then you see how the pathogen starts here and starts killing the different seedlings. Yeah? And then you have the control where all the plants are healthy and happy. Yeah? The one inoculated with Rhizoc, plants are dead, almost all. And then you have the one that is inoculated with your strain. So this is a good example for you, maybe, for your system. I don't know if it could work for you. And then what do we see here? Well, in some of the Burkholdeриas, we could see that the pathogen could not go farther. So for that, that's plant protection. Is that clear for all of us? Yes. Good. So then, well, here it's a summary of some of the experiments that I did using this system. And here you have the disease incidence, and in the x-axis you have the different strains. So it was quite nice because most of the strains were able in vivo, yeah, to protect plants against the pathogen. There were some that were more efficient than others, but in general, all of them were able to protect. So that's quite nice result, and then I was okay. In vivo, they have some kind of activity, okay? So then my message here is that, okay, so if you see a strain that in vitro is not doing much or nothing, it could be that in vivo it's doing, okay? Good. Then we move to the next step, because I told you that via diffusible compounds there was no activity. But then we saw that via volatile there was activity. And I think this, you did it yesterday. This is what we call the sandwich bioassay. So you take two bottom plates, yeah? And then in the bottom you grow the bacteria, and in the top you grow the fungus. Yes? Yes? Good. And then this was the result. So I had 5 different Burkholderia strains, species. And then here in the y-axis you have the different species, and then in the x-axis you have the fungal growth in centimeters. So here you have a picture of how it looks like. So here the bacteria in the bottom, and then here the fungus. So you see how the fungus, in this case was Rhizoctena solani, it's growing in radial growth, yeah, and then until covers the whole plate. What was extremely interesting for us was that, well, Paraburkholderia graminis, because they changed the taxonomy not so long ago. Paraburkholderia graminis, 1 out of the 5 different species, was able to inhibit the growth of the pathogen via volatile. You can see here in this picture. I hope you all can see. Yeah? Good. And then the others not. That's a quite nice result. What to do next? Because Josh told me, Vic, what to do now? What do we do to find the mechanism? We know that it's via volatile, but we need to do something else. I cannot hear, guys, sorry. Yeah, that's one thing. Yeah, exactly. So one thing is identification of volatiles. And while you do the identification of the volatiles, what can you do in the meantime? Yeah, we are really interested in pinpointing the genes involved in this phenotype. genome sequencing. We have 5 isolates, right? So while we do the volatile experiments, we are going to do the genome sequencing because, of course, we have 5 bacteria that don't have the phenotype, 4 bacteria that don't have the phenotype, and 1 that has the phenotype. So that could give us a lot of information. Yeah. So well, we sequence the 5 genomes. These are this is the table that you can make when you sequence a genome. You look at the number of chromosomes because Burkholderia is a quite complex genera. So it can have 3 chromosomes and several megaplasmids. Yeah. So then ours were okay. So we have 2, 1, 2, 1, 3 chromosomes, 1 plasmid, and so on. In terms of the size, they were more or less the same size, except Burkholderia hospita, that was extremely big, yeah, around 10 megabases. And the good thing is that, well, for some of them we had several draft genomes, so we could do some, we could have a reference, and for some of them were the first genome that was sequenced. Then next thing, well, while we were sequencing, what we did, we were looking at the metabolomics, at the volatomics. So we were looking at the volatile organic compounds produced by this strain. I hope that you can see here. You see? It's a bit white for me here. Well, so how does it work? Well, you have these columns that you put it on top of the plate. It's a special plate that we had in the lab, and then you grow your bacteria here, and then basically the bacteria will produce the volatiles, yeah? Okay? Will produce the volatiles, and then we will capture them in this column. So then now we take out the columns, and then we go to the GCMS, because we are doing gas chromatography, and then we will measure the volatiles. What was the result? Well, you have it already here. So I did it for all the strains that I had in my collection, all the species, the 5 of them. And then you can see already that Paraburkholderia graminis, the one that was causing this inhibition of the growth of the fungus, had a quite different profile. So here we are doing a kind of beta diversity. We are looking at different treatments. Yeah? So we have Burkholderia graminis, Paraburkholderia terricola, and all the others, and the control that was PDA, and they were all here, and then Paraburkholderia graminis had a significantly different profile than the others. What do you do next based on this metabolomics data? That it's basically a data frame with a lot of volatile compounds and abundance. Yeah? Kind of. So then we infer more or less the compounds that could be involved, yeah, or that were significantly produced when we compare Paraburkholderia graminis with all the others. So we wanted to know which volatiles were unique, yeah, to Paraburkholderia graminis. So we detected several ones. Yeah? So when we did the analysis, here I'm showing you a heat map. So you can also see here that the profile from Paraburkholderia graminis in kind of orange was completely different than the others. And then we could see that most of the volatiles that were differentially produced by Paraburkholderia graminis were volatiles derived from the sulfur metabolism. That's already giving some hints, right? So what do we do next? Huh? Exactly. And what will be our focus in the comparative genomics? Exactly. So the idea is that, well, first of all, yeah, so we we call pan-core genome analysis. Do you know what is the pan? Guillermo will explain in the afternoon, but the pan is all the genes in a given data set. And the core are the genes that are shared among all our bacteria. Okay? So we did a pan-core genome analysis. But for the pan-core genome analysis, what is our main interest? When we do the pan-core genome analysis, you get the genes that are core, so the ones that are in all our Burkholderia strains, and then the ones that are shared between 2, 3, or whatever. And, well, we get also the unique ones. So which gene set should we look here to find our genes of interest? In the unique genes from Burkholderia graminis, right? Well, that's what we did, and then we found around 10, 20 genes that were involved based on the annotation, in the sulfur metabolism. So we found 10 or 20 genes. What was the next? I generated mutants in some of them. Some others I couldn't make the mutants because maybe there were genes that were essential for the life of the bacteria, but I managed to make several mutants. And this was the result. So I'm showing you here, again, a plot, a PCA plot, where we can see the general overview of the metabolome of the 3 different bacteria. I mean, the Burkholderia graminis, that is the control, and the 2 mutants that we generated. So I'm not going to name the mutants. But what was more interesting is that when we measure the volatiles from the mutants, we could see that indeed some of the sulfur-derived compounds or were gone from our analysis from our results, or where the production was reduced. But even more important, what we did was in vitro and in vivo analysis with the experiments with the fricandel assays. So what we did here, for example, in the in vitro, we did the sandwich assay, and then we see that Paraburkholderia graminis is still inhibiting the growth of the fungus. You can see here the different treatments, the different bacteria in the y-axis, and in the x-axis you have the radial growth. And then the 2 mutants that we generated, they were not able anymore to inhibit the growth of the fungus. But what happened when we moved to in vivo? The fricandel assay, exactly the same. So we were able to see that indeed Paraburkholderia graminis was able to protect the plant against the fungus, but the 2 mutants lost the ability. Okay? Any question? Very clear. Nice. Okay, well, this was an example of how to use comparative genomics to identify your phenotype of interest, the genes involved in your phenotype of interest. I hope that it was clear, the example. But anyway, if you have any question, please. Yeah, there is a question there. Nice. Is there a microphone? Yeah, you're coming. Yeah.
So in one of your initial slides, you told that there was no any effect of that particular bacterium on in vitro assay, but it had some activity in the in vivo assay, right?
Can you repeat the question? I didn't get
So
it.
the bacteria which you have isolated, like had shown like not much effect against the fungi in vitro assay,
Yeah.
but it had some effect in vivo assay.
Yeah.
Right? So what may be the possible reason that like it could not show any effect in vitro? So, does that mean like it requires some signal from the plant to
Yeah. Yeah, that's an amazing question. I mean, yeah, it could be that it requires the two or the three-way interaction. Maybe not only needs the plant, but also the fungus to be present. So yeah, I don't know, but most likely there are some signals. Maybe I think Josh will expand more on Monday about the crying for help and so on, but maybe when the plant is under attack, it releases some molecules that activate these compounds in high concentration to inhibit the growth of the fungus. Yeah, but that's an excellent question. Yeah.
So because we generally screen out, we think that the one which is performing better in plate assay, in vitro assay, will perform in like plant bioassay.
Yeah, but I don't know whether that's a good. I mean, it's a fast way of doing it, but it could be that some bacteria are also active when they are in vivo, so together with the plant. So then you will be missing those ones.
Here the mechanism is different. Here they are focusing on vivos. Here the normal screening it was not showing any. But when they specifically started doing for vivos, then it is showing a disease.
The focus was on VOC, volatile organic compounds, and actually many compounds can also come from the secretome. Microorganisms can evolve
I cannot.
compounds from in the secretome. Not only VOCs, but also it can come from the
Well,
diffusible compounds.
Via diffusible compounds, we didn't see much activity, to be honest. So mainly it was volatile. So when we were looking at the diffusible compounds, there was only one of the strains that was Burkholderia hospita that was giving a bit of activity, and not against rhizoc. It was given to another fungus. So then that's why at some point I decided to move to volatile. Yeah, because before I had already the information from the bioassays, because I said, okay, well, they should be doing something. So then that's why I moved to the bioassays first, yeah, and then later on I found out that indeed it was volatile. Yeah.
Bacterial inhibition of fungal organism is more due to volatiles.
Yeah. Yeah. There is another question there. Sorry, there is another question. Yep.
And I wanted to ask that are these volatile compounds always released by bacteria? That means they are constitutive in nature, or they are induced only with some pathogens?
Yeah, very good question. I don't know. Could be that they are induced by the pathogen. Maybe they are induced by the plant. We could detect them, although at low concentrations. We could detect them in vitro at the end. But also it's quite specific from the media that you use. So in this case, I think we were using 1,5-PDA if my memory is still okay. Yeah, but it will depend a lot, on that, indeed. Yeah, so maybe it's activated through some compound of the plant, or I don't know. Yeah. There is another question in the back. Yeah, thank you.
So you told that most of the pathogeny or antagonistic activity by bacteria will be shown by volatile organic compounds, not because of secretomes.
Excuse me, can you repeat the question? I didn't get it.
So you told that the inhibition of pathogen by bacteria is mainly because of volatile organic compounds, not because of secretomes.
Well, no, I didn't say that. No, 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 no. No, no. In my case, for this specific example that I gave you, yeah, the activity was through volatile. Okay. Yeah, but that doesn't mean that other bacteria will do also through volatile. That, yeah. Sorry if I didn't explain that well. Yeah, sorry. Yeah. There is another question there, please. Yeah, thank you.
Sir, like these volatile compounds, whether they, in case of a plant system, whether they will be antagonistic to other bacteria which is of economic importance.
You are asking if they could affect other bacteria? Yeah, actually there are quite a lot of studies.
That bacteria is of importance or it is.
That's, yeah, I don't know. Yeah, so but for sure I didn't test, but I know that there are some works from, for example, Paulina or Chun-Min Ryu, Paulina Garbeva and Chun-Min Ryu, where they look what's the effect of bacterial volatiles in other bacteria. And they saw, for example, that in E. coli, well, E. coli, whether is that happening in the rhizosphere or even in the endosphere, for sure, I am sure. Yeah, the rhizosphere and the endosphere, it's a battle. So there are so many bacteria there, and they will be producing compounds against each other because they are competing at the end for the niche.
So another question is like whether is any alternative for this comparative.
You mean alternative in which sense? Like not command line, or sorry? So like, you mean if there is a web page to do comparative genomics? You mean, do you mean if there is a web page to do comparative genomics?
More philosophical.
Sorry, I didn't get the question.
Sir, after the sequencing, like in your research, you went for the comparative genomics. Sir, whether there was any alternative, like.
Alternative way to get to where I got without genome sequencing.
No, sir, after the genome sequencing.
She's asking you the very purpose of doing genome sequencing and comparison.
Well, I mean, if I would not do comparative genomics, I think I would not get into the specific genes. And I think what is more important is that you have 4 bacteria or 4, 5, 10, whatever, that don't show the activity, and 1, 2, or 3 that show the activity. So then you can compare their genomes and then try to see what is common, what is not common, what is unique for your strains, what is shared, and then you can pinpoint those genes. In the afternoon, we will see an example, and yeah, we will run it together.
Sir, like with comparing if some of the other strains are left out, then like you cannot take in Burkholderia only there are so many species.
There.
One genus. So like you can compare all those.
Yes, you can compare. Actually, the PhD of Guillermo, it's generating a comparative genomics workflow, an automated one that you just put all the genomes and then you find the genes that you are interested in. So, for example, he took more than 15,000 Burkholderia that are out there, and then we were able, for example, to identify genes involved in virulence against rice. Yeah, so that's more. Is that answering your question? It's time consuming, but imagine that you have the pre-computed data and you are working with Burkholderia, and then you just upload your Burkholderia to that database, and then you get the genes of interest based on the analysis. That would be amazing, right?
Then how will you
metagenomics. So next step is to do the same, but comparative metagenomics. Yeah? Excuse me?
Can you extend this your work of comparative genomics into functional genomics?
Like functional genomes, what do you mean exactly by functional genomes? Oh, what do you mean by functional genomes?
Here we have identified the gene sequences in the comparative genomics.
Yeah.
We can go for functional genomics to identify the metabolites.
To include transcriptomics. Yeah. So, I mean, there is no tool to do that, but yeah, so in the PhD of Guillermo, we are also doing that. So we are coupling transcriptomics and comparative genomics together to not only have the genes that are, most likely doing the phenotype that you are interested in, but also how is the transcription of those genes. So we are kind of, imagine that you are, starting with a lot of bacteria, and then you are, this is like a funnel. You know, you are trying to pinpoint exactly and spend less time as possible to get into where you want to go, to identify your genes involved in that function. I think Josh wants to say something.
So I think it's an excellent question. So the question actually was, do you really need comparative genomics to find the genes of interest involved in the activity that you see, right? The answer is no. And I will give you an example, but in fact it's yes. But I will start with so suppose that each of you, you have a super interesting whatever microbe that gives a beautiful activity, and you're interested, okay, what genes are involved, what metabolites are involved, how you regulate it, right, all that stuff. Then you can go for the, and I'm taking the microphone because I come from that sort of like generation. You can take the strain, and do, for example, a random mutagenesis. So you can generate many, random mutants, and then test each of these random mutants. We have done that. Each of these random mutants, you can test them in the system, for example, the sort of system that Victor showed, or in vitro assay, etc. And then try to find those ones that have lost the activity or have a reduced activity, right? And that will bring you to a sort of like a genomic overview of which genes contribute to the phenotype or not. But you can see the but coming. But just for an example, so Betaproteobacteria genomes of 6, 7, 8 megabases. Now to get full coverage, with a random mutagenesis of all the genes in the genome to pinpoint. In this case, so for Pseudomonas, this is relatively smaller. The Streptomyces genomes I mentioned, it's 10 megabase. You need to screen, and I'm probably off here, but you can calculate that. You need to screen at least 10,000 or 20,000 random mutants in an assay to have a full coverage of your genome, right? So in that sense, and that's the beauty of this, is that through comparative genomics with strains with genomes that do not provide that phenotype, you can have a much more targeted approach. There is another but here again. So it might be that you're probably going to look for genes that are unique in the strain that shows the activity and are absent in the ones that do not have the activity. That's sort of like, and in many cases you will find it. It can also be that sometimes genes involved in a specific activity are present in also the ones that do not show the activity, but they are regulated differently. And that's why you don't see the activity. Then transcriptomic comes in. Yeah, right? There's always a but. So there was one of my PhD students, he had this it was actually a super simple assay on a plate with plants and microbes, and I'll show some slides next week. Then you want growth, phenotype on the roots or root architecture, and he wanted to know which genes are important for this phenotype. And he said, we also want to do transcriptomic analysis. And I said, well, is that really a good idea? And he said, yeah, we're going to do this. So anyway, I said, go, do it. So we did it. And what happens then, it is, but the timing of the sampling, to do the transcriptome analysis is going to be crucial. He was too late. And then it's like, sort of like a fragmentation bomb, because all the genes, or many of the genes in the genome were expressed. But of course have nothing to do with the phenotype. So, yeah, so there's always pros and cons to each approach. I think, with the tools that we have and all the genomic information that we have now, if you can go to comparative genomics, it will help you to sort of like zoom in much more faster to the groups of genes that might be of interest. Yes, you will miss, for sure. But once you have some core genes that contribute to that phenotype, you can start looking at which genes are co-regulated, with those genes that are important, and that will bring you then also to basically the full set of genes that are required. Yeah. So excellent.
Thank you, Jos. Yeah, I also have done random mutagenesis in the past, and I am not. I'm younger. Let's leave it there. But then, yeah, it's really time-consuming, and it's a lot of work. But yeah, it's another approach if you don't want to do comparative genomics. But yeah, it will take some time. Okay. I don't know if there are more questions, but then maybe we can summarize, a bit. Yeah, I have also something else to. Yes. So, well, this is the summary of today. So you will get these slides. I hope that you learned today some of the different annotation tools that are out there, a bit of the basis, how general and specific they are. You know a bit of secondary metabolite and a bit more. The key basis of comparative genomics, and then comparing metagenomes can reveal that we could not find using only one genome. Okay, so what I mean here is that when you are doing genome sequencing, one genome, it's not going to give you much information. But if you have the genome that you want to sequence, you want to target, plus others that are not doing the function, then that will give you much more information. Well, and this is more a thought than. So I hope that you agree with me that comparative genomics methods are changing the biological research, okay? So we are getting so much data, not only genomes, but also metagenomes, and we basically need to find new ways to extract the information and, to get where we want to get. Okay? Thank you very much. And now, before we go, we have some notes. So I also want you to, let's see if we can do it. Okay. Okay, so if you go to the browser, so if you put Cytoscape, in Google, you will get the first link that you get, you click on, and then you will get into this page. So if you can, please download and install this program here. Okay, so if we have time, I would like to show you in the afternoon, in like maybe 15, 20, 30 minutes, how to make a network. Okay? Good. Guillermo. Yeah. Aquí tiene el micrófono. ¿Tú has puesto también mis cosas? ¿Has puesto mis cosas? Te manda un email. Ahora después lo vemos. No pasa nada.
Comparative Genomics Day 4, and you will just have to download these two files. You don't need to download the extra data. There I have outputs of some of the softwares that we use in the practical, but you don't need to run them. You just need these two files, the Comparative Genomics RMD and the comparative-genomics.r data. So just create a folder in your desktop and download that two files. All good to download it? Thumbs up to manage? Exactly. Anyway, Victor will show during the practical how it works if you don't manage to install it. Victor, say that if they cannot download, it's okay, that they can follow you in there.
So, because, so if you cannot download it because it's too big, what we can do is, we can transfer with a stick, with a USB, once and download. Exactly, but anyway, what I will do, I will show in the screen how to do it, and then you can take your own notes, and then for the ones that the neighbor has it installed, so you can go to e-market, okay? So
tell them to download this first.
But this one you need to download. That's very important. The other one is optional. But this one is mandatory, yeah? If you don't download this one, you don't get the diploma. Right, is optional.
I can share.
Is there? Oh. Whoop. No worries.
So if it's 5 minutes, it's good. If it's 4 hours, no.
No.
Yeah, more than 4 hours.
3 hours is okay, right? Big. ¿Cómo son? Bueno, si me los has mandado por email no deben ser grandes los archivos, ¿no?
No, son
vale.
súper pequeños.
Entonces los subiré luego y si quieres cuando cambiemos
Y ya mientras ellos lo descargan yo me...
setup y todo. Perfecto. Muy bien la lecture, ¿eh? ¿Te ha gustado? Sí, sí, me ha gustado bastante. Porque la parte de explicar las bases de datos y eso puede ponerse muy aburrido, pero lo has explicado bastante bien. Ojalá me lo hubieran explicado así, a mí. Sí. Realmente es muy aburrido. Es esta. Is downloading here? Ah, nice. Then it's easy to install. It's not like an R package. It's just next, and install.
For the ones that were able to download Cytoscape, it's like to install. Okay, so shouldn't be a big problem, I guess. And if there is any problem, just cancel the installation. You just double clicking, and it will open the installation software. Okay?
So while downloading, it stopped and it's coming like failed needs authorization.
What?
So
What? That's weird. You don't have authorization in your own lab? Try to download it maybe again. Like try to restart. Then go to the link again maybe. And if not, some people had it already in a USB stick. So, but anyway, it will be okay if you cannot install it and anything. But this is a program that is software that is good to have for visualization and everything. It's really nice. So try, but if not,
For what do you use it?
I'm using for constant annotation, grouping, the flag.
Beautiful, nice.
we don't have Java.
But it also said
Oh.
that Java is
What?
It's
automatically
gonna be a
downloaded.
big issue.
Java.
Java.
Does it give you the option to install it?
But then install it too. I mean, it should install Java if you don't have it.
Actually, it's very useful to have
Yeah, Java is...
Java is in our
So maybe when you try to install Cytoscape, it will ask you to install Java. Install, because many of the programs that we use needs Java. So just install it. I hope it works.
How many hours?
It is not showing that.
47
Well, it's going.
If not, I will show it on the screen. I think it will be nice for
Them to play around. Yeah.
them to know.
Josh, after lunch, are you gonna do first the wet lab? Okay.
There's so much data.
think what we are going to do for the
I have some slides.
we really need to think. Yeah.
We need to think.
I think we need to maybe get a differential, for the what we have, we need to do a differential abundance thing at the functional level.
Which one?
Your first part of the lecture was difficult.
Yeah, it's
Because it's, CAG, COG, and it keeps on
how
coming.
how to do it differently. It's just
Yeah, but I think he did it pretty good. I told him like I wish that my teachers in the Master of Bioinformatics because it's like that. It's like don't trust
so
them.
but it's not that it's not well presented. It has nothing to do, but it's just the amount of information.
it's a lot. It's a lot.
And it's
I
boring.
I thought we were done with the databases, like but there's more, and there's more, and there's more. Yeah.
Well, but I
that's
I
the,
tried to stop, and also the table that they couldn't see.
So I think if we want to get the best out of this afternoon, and I think we have done that, is that we gotta have it super structured, and we also need to make sure that we're not going to go all the way down to 5:30, but that we preferably end at 4:30, right? Should be feasible, huh? Yeah.
I mean, depending how the network thing. I think
No.
my
If
thing
it's-
would be max
If
1
it's
hour.
4:30 and the network is not done, we don't do network.
We do it on Monday.
Yeah.
If possible. If possible. Yeah.
Or on Tuesday.
Yeah. Or on Tuesday, and then they
Oh,
can
no,
download.
Tuesday is late.
Yeah. Tuesday.
Okay, with that we will close the forenoon session. We'll break for lunch now. Post lunch, we are going to have that bioassay, what we planned earlier. So you complete that bioassay,